Okay, so let's get started. Welcome to the HEAS Genomics seminar. We are happy to have Stephan Schiffels here from the Max Planck Institute in Leipzig. He is a population geneticist, which originally was a physicist. He studied physics, and he afterwards did a postdoc at the Sanger Institute. He is a group leader at the Max Planck Institute since 2015, first in Vienna, then in Leipzig. And he studies a lot of different aspects of computational modeling, ancient DNA, including theory, but also he will tell us something about human mobility and population structure. So the stage is yours.
Thank you. Thank you so much for inviting me. It's a pleasure to be here. I have actually several connections to Vienna now, and you can see that in the setup of my talk, I've tried to sort of put as much as possible into this talk, which may be a mistake. So you can tell me afterwards whether it worked or not. But I sort of, I think mobility is really a nice umbrella for this talk. And I want to really just in the beginning give a little bit of context, that we can talk about human mobility really on very, very different scales. We can talk about human evolution, where do humans come from on a sort of really global scale. We can look at continental scale and subcontinental scale relationships between humans and what that tells us about ancient mobility or present mobility or, mobility throughout human history. And then also there's this kind of smallest scale when we really talk about individual communities with each other. And I'm going to talk about all of these, but there's going to be a very heavy bias on this intermediate scale because I actually think that's the scale that is sort of population genetically the most interesting one, where also ancient DNA can provide the most additional means. But this most recent work here comes last, and I'm particularly glad that some colleagues from the Natural History Museum of Vienna are here, which this project is about. I'll tell you about that in a second. I just want to start to set the stage with the sort of the first scale, the biggest scale. For most here, or for hopefully maybe all of you, which will not be news, that the vast majority of human ancestry, actually more than 95%, I think, on average, follows this kind of overall picture. So we have a global evolution, or a Pan-African human evolution, where humans emerged sometime 200,000, 300,000 years in Africa. I need to be careful what I say because Tom and Katarina are here. I'm sure you have lots of details to add to this, but more or less, this is the rough picture. And of course, then humans ended up in all the places in the world. My own contribution to this research is many years old. I just want to sort of briefly mention it. One way to sort of visualize this, and this is a particularly simple visualization, and I really like simple visualizations. Most of my visualizations are not simple, but this is actually made by Ian Matthews, and this is just heterozygosity of all modern human populations as they have been available some years ago, plotted, and this Ian came up with this sort of a little bit cheekily as distance to Addis Ababa. Of course, Addis Ababa is not the single cradle of humankind, but still sort of it's useful to plot it as some kind of distance to Africa. And you see, of course, a pattern here that the closer you are to Africa, the higher your genetic diversity is. And as you further go down, as you further go away from Africa, find populations today away from Africa, the smaller diversity is. Of course, this is not the only picture. You can refine this, and this is where my own contribution comes in. 2014, we published a method that looked at this not as a single point per individual, but as a whole curve per individual. So looking at effective population sizes through time, which I like to kind of show. Sorry, I don't see the next slide coming up here, so this is a little bit annoying. So there will be surprises in this talk. So the next slide is apparently this one. So the reason for this kind of for this successive decrease of heterozygosity is, of course, bottlenecks involved in how humans settled one continent after the other and one place after the other, everyone involved with bottlenecks. And one way to visualize this is, for example, a method that we developed in 2012 to 2014 and published in 2014 called MSMC. And I just wanted to briefly mention this here because it's my own contribution to this rather broad and global story. So if you sort of lay out human evolution on this kind of rather bold x-axis scale, going all the way from a few thousand years ago to, a couple million years ago. And I've put some landmarks here, divergence from chimps sometime, but several million years ago, the earliest fossils that are unambiguously modern human, about 300,000 years ago from Morocco. And then, of course, the more recent evidences for human culture in the last couple of thousand years ago. Then what you see is if you now model every individual, not just as a single point, as a single heterozygosity estimate, but as a function through time, you see these kind of this phenomenon that, human evolutionary population size, effective population size was rather similar in multiple populations from Europe, Asia, and Africa, and then started to diverge sometimes about 300,000 years ago into a stem inside of Africa and into stem outside of Africa. And remarkably, if you add, you can now ask what happened here in this time point. And of course, we don't know really. It looks like effective population size were low. This looks like a bottleneck. And it just so happens that this is about the time point that we now schedule, or that, also we have the archaeological evidence that that humans started to diverge outside of Africa and stay there. There were previous movements out of Africa, but this was sort of maybe the most important one from a present-day genetic perspective. And remarkably, if you add more curves, you see more details. For example, the South American, Native American populations exhibiting this later bottleneck around 20,000 years ago. Again, we don't know what happened, but this happens to be the time point when we know that the first humans started settling in North America, at the time crossing the land bridge because ocean was much lower, so there was a land bridge between Asia and America. This was about the time. The true story here is also extraordinarily complicated. I've worked a little bit on this, but there were ice sheets there first, kind of blocking the corridor down to America, and then at some point they opened and so on. So there's lots of details here to uncover, but roughly the story goes like this. And remarkably, if you add more populations to this, you see that human evolution is really sort of follows this kind of common stem here. There's again lots of details to this uncovered in recent work, but roughly most of our ancestry follows this kind of one stem, and then there's this one out of Africa branch and multiple branches of extant populations today in Africa that follows these bottlenecks. And I just like this kind of, like starting with this work because it gives this global idea that, of course, there is a hallmark of this global mobility in our genomes, which was one of the things that really drew me into the field. The first time that I saw these curves, I had the feeling, wow, okay, so this is something that is real, that sort of looks like there's a real trace of the past in present-day human genomes. And this story is encapsulated in these genomes. And ultimately, I think it's a story of mobility that is present in our genomes. And sort of that sets the first stage. I want to sort of start the next chapter with a quote that I take credit of, but that should give full credit to my now postdoc and then PhD student Clemens Schmid, who started our paper on mobility in 2023, published in 2023, with this beautiful language. All human behavior is spatial behavior, and spatial perception and interaction are deeply rooted in the human mind. Understanding movements in space, mobility, on different orders of magnitude is therefore a major component of understanding human behavior throughout history, from the Iceman's quest to the Ötztal Alps to the Viking expansion, and maybe eventually humankind's escape to the stars. We managed to get this actually through peer review, so I think we are the first ancient DNA paper who mentions space travel in an academic journal, not just space travel, but actually interstellar space travel. The only remark that the reviewers had is that it's rather dystopian to put this escape here. So they made us change this to journey. Sorry, this cross is wrong. It should be, we needed to change escape to journey. This is badly layouted. Anyway. So Clemens and I really looked deeply into mobility, and I want to start with some examples that sort of exemplify what is the deal here. Why are we looking at this? So I just give some examples of research projects in recent years that sort of uncovered this kind of central concept that we can use genetic ancestry to trace where people come from. This first example is from a project published in 2016, not by my group, but from colleagues from Dublin, Rui Martinano et al. They looked at a bunch of at a couple of burials from Roman time York. Like York was a major Roman city. It was one of the places where the Roman emperor was crowned, where a large fraction of the Roman army was. This is not a small provincial town. York was for times really a major place in the Roman Empire, and they had 6 or 8 burials from this that were associated with Roman soldiers. And if you plot, and this is what they did at the time, they kind of laid out a map of human diversity, present-day human diversity, that is. So every point here is a present-day living person, or I'm not sure whether they're still alive, but they're sort of from whatever the 20th century. And every point here is a genome, and they are put out in this kind of map, which is called the principal component analysis, where the distance between the points roughly matches the distance in genetic terms. Okay, so things that are far away are also genetically a little bit more distantly related in terms of allele frequencies. So we can use this kind of map to map out where these ancient genomes now fall. And you see that most of these ancient genomes from York, and there was not just Roman soldiers, there also had one individual from the early medieval period, so-called early Anglo-Saxon period, and one from the and one from the Iron Age, and they all plot in this kind of Central European place where more or less modern Britons are also situated. So no big surprises there, but there was one individual who plotted straight into present-day Jordan, Palestine, Syria. And of course, their conclusion was rather straightforward, and probably we all would have made that same conclusion, that probably that individual or their immediate parents must have traveled from here to here, right? So the central idea here is, and I'm coming back to this, that a shift in genetic map signifies a shift in physical space, right? That's a rather important conclusion. And it needs to be justified population genetically, and it can be justified by the insight that most human diversity in the last couple of thousand years is driven by migration and not so much by drift. So you should not expect major changes happening in this genetic space purely based on isolation and drift, but any changes, in particular on such a PCA where we use projection methodology, so basically we project ancient samples onto modern Eurasian diversity. In such a map, changes in genetic map can only be explained by changes in physical space. Okay, so that's the central paradigm, and I'm coming back to that. You see this in all of the examples. The second example is an example from my own research where we have in a project where we've looked at Anglo-Saxon England. I'll come to that project in a second, but this is a remarkable outlier. One individual stood out from Kent, from a field called Updown Estry. It's a 12-year-old girl, and our archaeologists called this project the Updown Girl Project. And this girl is rather remarkable in terms of genetic ancestry because she falls a quarter way towards present-day Africa. Okay, so where all kind of her relatives, even her immediate relatives that we have found on the graveyard, plot all into European diversity, she's the only one falling a quarter out into Africa. And of course, the interpretation is not so much of a surprise, but it's clear that someone in her immediate family must have traveled from sub-Saharan Africa to Britain, and at the time, at like 2,000 years ago. Again, not terribly surprising, but kind of nice to see an example of this. And indeed, there's genetic evidence. You know, the brighter the color here in yellow, the more likely this kind of ancestor comes from. So probably somewhere West Africa. Even when we actually decompose the genome, we could actually find places in her genome that are unambiguously sub-Saharan African origin and other places that are European origin. So we actually see the traces in her genome that she must be, someone with 1 or 2 generations back in her family pedigree having African integration. We computed this to be the most likely tree, so probably her paternal grandfather was African. Again, a signal in genetic space signifies a signal in physical space. That's the idea, right? The last example and you can see that I'm sort of putting out this umbrella just as a trick to kind of show you all of my research. So this is the much bigger project that we published at the time, not necessarily under the explicit umbrella of mobility, but it turns out to be a story of mobility. So we looked at Anglo-Saxon England. We've sequenced like hundreds of, I think 500 samples overall from northern Germany, northwest, sort of the North Sea coast countries, and Britain. At the time, I think the second largest study for ancient DNA. You can see lots of samples here in Britain, covering 400 to 800 or 700 in Britain, in Germany, and in Denmark, and in Netherlands. We have a little bit of a broader spread. Dating isn't always perfect, but overall, this gave us a good picture of what was going on at the time. And what we find, I can kind of show you this in this kind of comic slide succession now of plots. Just to orient you, this is a plot of Northwestern Europe today, where we're going all the way from Ireland through Wales, Scotland, England to Netherlands, and then following the V shape here, Netherlands, Germany, Denmark, Norway, and Sweden. So we have kind of a Scandinavian edge here and a, dare I say, Celtic edge here, but Celtic here understood in terms of languages as they've spoken today. And then in between we have these other countries. Okay, so that's the background here in gray. And if we plot our ancient samples from early medieval England and Germany and Denmark and Sweden onto these plots in temporal order, we first of all, of course, this is not just our data, also previous data from England Bronze Age. We see kind of this cluster following more or less where present day Celtic speakers are. And somewhat overlapping with England as well. It doesn't change much in the Iron Age. This was remarkable to us. There's more subtle studies now that show that, yes, there was actually a movement. It is very subtle, but if you model it right, you can see it. There was an influx actually from France, which is not shown here on this PCA. We've done other tests that show this as well. But for the story here, it's only important that there was not any change in England from the Bronze to Iron Age with respect to continental North Sea Europe. This is quite remarkable. This shows 2,000 years of continuity, or at least 2,000 years of absence of North Sea gene flow. Because I later got asked a lot, like, well, isn't it normal that you have something like the Anglo-Saxon migration? Because, you know, England and the North Sea, that's not hard to cross. Well, it wasn't crossed in large scale, right? There was 2,000 years of basically no migration going on between the continent and England, at least into that direction. Over the Channel, it was different, but over the North Sea, there wasn't much contact up until the early medieval period. Sorry, this is one intermediate step. These are the early medieval samples from Germany, Netherlands, Sweden. So this is not England, this is continental Europe in the early medieval time period. So it shows that while we're projecting stuff on modern day diversity, we can still reason about ancient processes, right? Because we're projecting everything. We're even projecting the putative source region of this migration. And then if we plot England, of course, we see this massive shift from the blue region over to the yellow region, okay? So during early medieval England, there was a massive shift. All the communities that we studied had something like 75% continental ancestry in early medieval England, starting from the 5th century onwards. An absolute dramatic shift, and something that archaeologists actually encourage us to call a mass migration. It's a little bit of a weird political term today, but in the scholarly debate, it is justified because that was sort of one of the terms that was used to describe this process. The other extreme was that it was just sort of a political elite transfer, that people actually changed language, but that it wasn't a big migration. I think this clearly shows that there was a massive migration from the continent into England during the early medieval time period. We can also model this a little bit more opinionated with two ancestries here in blue, the Iron Age, Bronze Age component, and in red, the continental component. And we see this massive rise here in the early medieval time period, which is not the case in the Iron Age, at least not at the same scale. And we can also, and anticipating another method that I'm going to present later, MOBEST, which let us estimate the origin of these samples. And you can see that we can nicely plot them, that we can infer. These are all English samples, mind you, right? These are all samples buried in England, but we're plotting here the likely origin of these samples based on a machine learning method using a large amount of ancient samples. That's the next chapter. And you can see that we got the source region pretty covered, also where historical sources mostly put it. Okay, so we were thinking like all of these examples were meant to kind of show you the core idea that changes in this kind of genetic space show changes in physical space. So we can use genetic space systematically, or shifts in genetic space systematically, to infer mobility. And that was what we did with this project from Clemens Schmid, which I'm now going to present. So Clemens at the time compiled a big data set of everything that's been published up at that time about Western Eurasia. Covering nicely space, of course, with a little bit of a wiggle here. Of course, lots of these samples are from the same site, so you would see massive overplotting. So we have put a little bit jitter here to plot them nicely, but you can see that we're covering a lot of the area quite nicely, of course, with some gaps. And also temporally, we have a relatively good coverage in the different regions here through time for the last couple of thousand years. Again, some glaring gaps, but if you want to tell a story about the last couple of thousand years, like a global story of Western Eurasia, it seems like we are now at a place where we can actually do that. We may not have answers for every time period yet, but we can sort of try to kind of be global about this. The idea here is that we have a rather complicated problem, right? We have 2 spatial dimensions and 1 temporal dimensions. So that's already 3 dimensions that every single individual has. And now we have genetic space, which I'm going to go through in a second, which also adds more dimensions. So you have 5 and 6 and 7 dimensional space that you need to get kind of covered. Let me just briefly show you that on a genetic map, and this is, I'm quite proud of this plot, because this is not a projection plot on modern day diversity. This is entirely a multidimensional scaling plot on ancient genomes. To the connoisseurs here in the room, if I were to plot component 3 of this multidimensional space scaling plot, I would see shotgun versus capture. So it is sort of, it captures a temporal spatial temporal dimension very nicely. As you see, the oldest ones are here, the youngest ones are here, and so on. So, and there's also geographic clustering, but it is not easy to do this. And if you go to higher components, you will see that ancient DNA batch effects will dominate this kind of effect. But we also tried projection PCA. This is now for the experts here. Anyway, let me go through this real quick to show you that there is a pattern. So you see the oldest genomes, Mesolithic hunter-gatherers, all the way clustering here. You see then Neolithic Europe. So the first farmers in an entirely different place. Mind you, these 2 ancestries are about as different as present-day East Asians and Europeans. So is quite a genetic difference. We have another source here that played a big role in ancient DNA in the last couple of years, Pontic steppe herders, which had a big influence on European genetic diversity. And all of present-day Europe is now here in the middle. So this is clearly a mixture of these different sources, which has been well described. And we wanted to kind of try to make sense of this in an automatic way. We want to go beyond storytelling and beyond sort of looking at individual places like this Roman soldier or the Anglo-Saxon migration, which are targeted projects that look at very specific questions and then find answers for them. We want to kind of look agnostically at the entire data set and say, what can we say about human mobility from this data set? So I told you about these many dimensions. Here's one way not to visualize this, which is a 3D plot. And I'm showing this only for sort of conceptual for conceptual didactic purposes. So just to kind of remind you that we have 2 spatial dimensions here, X and Y, and 1 temporal dimensions here on the Z. So this is a space-time cube, if you will. And now color, we use color code for genetic ancestry. Okay, so the more yellow the samples are, the more they have of one type of ancestry, and the more greenish or bluish they have of another type of ancestry. You can do this with all of these dimensions. I've shown you 2 in the previous plot, component 1 and component 2. So that's already not visualizable. Now, the first step that we did is realize, well, there are lots of gaps, right? It's remarkably densely sampled, but as soon as you go to 3 dimensions, gaps will dominate the picture. It's sort of curse of dimensionality that hits you when you increase dimensions. You will suddenly have every data set is sparse in high dimensions, right? So we interpolated this. Again, not something to visualize in a paper or something, but just conceptually, we now filled this space-time cube with interpolations of this ancestry. That's the key idea here. Nothing fancy in this case. I mean, we spent months on this to get it right, but in principle, it's nothing fancy. It's called Gauss process regression. It's a method that sort of lets us relatively model-free interpolate space between these sparse points, and then For example, we can visualize this as this comic kind of comic strip kind of plot, where one component here is shown in the upper row and the other genetic component is on the lower row. And I just want to point out these visualizations or these interpolations, they do show major movements of human mobility. For example, you see that between 7,000 years and 5,000 years BC, suddenly all of Europe turns green, and in this case, dark purple. This is, of course, the Neolithic transition, the major movement of early Neolithic farmers coming to Europe. And another change here from 3000 to 1000 BC of, Europe again becoming a little bit more bluish, which is this kind of step transition. So the major stories are definitely there, but can we go beyond those? So the idea is now in this interpolated map, can we ask the question, this Roman soldier, which is buried in blue Britain, but has a green genome, right? That's what the color now signifies, a different ancestry in a place where it's unexpected. Can we now ask the computer, like, can you find us the most likely place of origin of this individual in a probabilistic, systematic way? And of course, we can do this by building a very simple probabilistic search where we simply say, where in the European map are we most closely to this kind of ancestry? Here's another visualization on how to do this. You can now plot sort of this ancestry landscape on the z-axis. It gets a little bit nerdy here, but essentially we're then cutting this. And there's a very intriguing parallelism to radiocarbon dating, by the way, where we also now have a certain measure point. This is now the genetic coordinate of this soldier, of this Roman soldier from York, and we're sort of asking where in the landscape are we most similar to this. This is a little bit similar to radiocarbon dating. It's no accident, by the way, that Clemens Schmid, my postdoc on this, comes from the field of computational archaeology, so he was very familiar with this kind of calculations. But ultimately, this let us draw a probabilistic map of origin for this individual. So based on his ancestry, we were able to plot out probabilistically where in space he comes from. I'm quite proud of the fact that we made this a proper probabilistic method, because this, of course, now allows lots of follow-up things that we can do with this. We can, for example, draw down likelihood models of certain movements and so on. So this is what we're currently following up with. But for now, it's only important that this method allows us to give a probabilistic map of origin for every sample. I want to give one more example that shows the power of this with a different individual, this time signifying one of the major transitions that happened in Europe from hunting and gathering towards sedentism and farming, here depicted by these artistic illustrations. So this Neolithic transition is one of the best described human cultural transition, at least archaeologically described, that there is. We have evidence for early pot making and sort of things that archaeologists call the Neolithic package. So things that appear together that convince us now that these people must have had this subsistence strategy and these other people must have had this subsistence strategy and this particular type of subsistence strategy that relate on farming and sedentism spread from Neolithic Anatolia and sort of the place that we now call the Fertile Crescent through Western Anatolia into Southeastern Europe, through Central Europe, and then later into Britain and Sweden, 4000 BC. And we can ask now, with an individual buried 5000 BC from Stuttgart, this happens to be even called Stuttgart. It's a weird signifier for the city, but of course it wasn't Stuttgart at the time. Anyway, this individual is one of Europe's, is a representative of the earliest cultures in Europe that represent this farming culture, which is called the LBK, the linear Bandkeramik. And this individual had an ancestry. If we now ask our method where, and this is to get a particular message across, where in 7500 calibrated BC would this ancestry be? And we can find out with the method, well, sure enough, this ancestry falls exactly where we map the origins of Neolithic farming. So in other words, this person's ancestors must have come from this place 2,500 years before this individual died. But now the interesting point about this method, because it's a truly spatio-temporal method, is it's not just a spatial method that sort of takes a bunch of training data and tells us where is this ancestry most likely. It is a method that actually lets us scan through time and ask where was this ancestry at a particular time. Lets us now sort of do this analysis through time. And you can now see, as I'm sort of scanning forward in time, the place of this ancestry changes. So suddenly in 6,600 BC, we're in Western Anatolia, and then switching over to Southeastern Europe around 6,000 BC. And then as you move time forward, you see that this ancestry becomes more and more local. And of course, this is now, this shows that this question, I mean, we're dabbling with this in political culture today as well, right? This question, where do you come from, is not a reasonable question to ask. You need to put a time horizon with this, right? It is very likely that this person was born near Stuttgart and that his parents were born near Stuttgart and his grandparents probably too. But his grandparents, they were probably born somewhere further southeast, right? So ancestral origin shifts through time, and getting this temporal dimension right is something that is still sort of bewildering me, because it means that we need to kind of clarify with every time we ask, where does this Roman soldier come from? At what time are we actually looking? Okay, so this is sort of some of the insights that this MOBEST method that we published, actually this is wrong, it's 2023, January, PNAS open. So I encourage you to have a look. There's one more kind of, how am I doing in time? Yeah, there's one more aspect of this that I want to kind of quickly get across. Now, the problem is, of course, we are sort of drowning in data, right? Once we had this method ready, we can now give a temporal origin map for 3,000 or so samples. We can now play this game with every single sample in our data set. And one way to summarize this is by a maximum probability vector. So, okay, so instead of kind of looking at all these kind of beautiful probabilistic map, so I'm shooting myself in the foot here. I praised the probabilistic aspect of this. Now I'm forgetting about all of this and simply say, let's just look at the maximum likelihood place. Oh, by the way, I wanted to get one interesting aspect about this Roman soldier. What I'm also quite proud of, that our method actually shows that, yes, he could have come from Syria, but he could also have come from Italy, because, of course, Italy at the time had a large influx of the Eastern Mediterranean, and there was actually a paper showing this by Roman time ancient DNA from Italy, which had this large influx from Eastern Mediterranean. So you can see that our method captures that correctly and rightfully says, of course, his parents or grandparents might have come from Rome, and their grandparents then might have come from the Middle East. So you see these multi-stage mobility somehow also reflected in here. But now we're sort of we're summarizing this all by a single point of origin vector, which has a direction and a length. And let us now summarize all of these genomes. So for example, here is the summary from Britain, where now on the y-axis, we're plotting the length of this origin vector. So the higher up you are, the more distant, the more evidence for long-range mobility you have. And you can see that there's two phases here, one in the early Neolithic period in Britain, or shortly after, and one in the early Bronze Age, signifying this step ancestry. We can also attach a direction to this arrow here in this color scale. So blue means probably somewhere from the east, and purple means somewhere from the south. So you can see, I mean, this is very, very rough summary now. This doesn't say anything about the actual places where people crossed the ocean to reach Britain. But you can see that overall the ancestry in the Neolithic expansion pointed more towards south, and the ancestry coming later in the steppe pointed more towards the east. Nothing of that is really new, but the summary, the kind of summary technique here is sort of is one way to summarize that. We haven't been able to do much more with these mobility curves. Originally, this was the motivation for this entire project. But I'm now much more happy with the fact that we can use individual ancestry probabilistic maps, and that's mostly what this method is now used for. But nevertheless, we can now also reason, and I encourage other people to do that, because I think there's lots of signal in this data that we haven't uncovered. But this is one way to sort of summarize it. And of course, we have these mobility curves for all of the European regions, for example, Britain and Ireland, Central Europe, and so on. And we write a little bit in the paper what we think these mean. Not trivial to interpret, but one way to summarize this into a comprehensive story of human mobility in the last couple of thousand years. Okay, so let's scale down one more step. And this now gives me the opportunity to present one of the more recent projects. And so this is a project that one of my postdocs pushed, really, Luka Papac. It's a large collaboration, again, with a lot of partners covering Southeastern Europe in particular. So this is in the umbrella of an ERC grant called Microscope, which had a particular focus on the European Iron Age. So the history right before the actual start of history. So it's proto-history, or you can call it. So the first millennium BC mostly. So we know a little bit about these places and about these people. We know a little bit about languages spoken. We know a little bit about mentions of historians, but we don't know much about real movements and so on. So this entire project is much larger than I can possibly present now. We have sort of, I think it's, we're analyzing 860 genomes, but new in this project is about 300, covering mostly the Iron Age. Here you see in this gold, covering mostly the first millennium. This is where we mostly fill up the landscape currently, and then try to sort of understand population movements and migrations and so on. So I'm not going to talk about all of these aspects today. I want to focus on one particular finding that my postdoc made that I'm still grappling with, and that also lets me think a little bit. Well, I'm going to show you. So what Luca did is he exploited a new technology or new methodology that's not new in DNA in general. It's called identity by descent, but in ancient DNA, it is certainly new. So the idea is that instead of looking at allele frequency shifts, so this is something I skipped over. It's a methodological a concept that in these methods that we used previously with PCA and so on, showing these genetic maps, these are all based on single point allele frequency changes between populations. Now we're looking at something that actually spans in the genome itself. So it happens to be the case that recent common ancestry, like, for example, these two individuals, they have a common ancestor 7 generations back. This is certainly too far to be knowingly related, right? These are not first cousins. These are not second cousins. These are maybe 5th or 6th or 7th cousins. I'm sure nobody here in the room knows their 5th and 6th and 7th cousins, right? This is distant relatedness, but which is nevertheless meaningful because it shows a relationship between these two individuals somewhere in the last couple of generations. And what happens is, if you have such a recent common ancestor, that you happen to have relatively long stretches of the genome that are similar. So we are looking not just as how similar is this genome and this genome in terms of overall allele frequency sharing. You're asking now, do we find blocks in the genome that are actually the same? If you have ever looked into these methods like 23andMe and so on, where you spit in a tube and send it to a company and they tell you, oh, your relatives are somewhere, 100 kilometers from here and so on. That's the technology they use. It's called IBD, identity by descent. So it's about finding fragments of the genome that are similar. And in ancient DNA, this has been exceedingly difficult because ancient DNA is noisy and you have tons of missing data and there's errors and you have ancient DNA damage, which creates weird artifacts. And to get this right, there's one brilliant PhD student actually in Nick Barton's group, who later then joined John November's group and who's now group leader in my department, luckily. In our department, and he developed this methodology for ancient DNA. It's called ANC-IBD. It's a new method that lets us look at IBD fragments across ancient genomes, and that opens an entire world of new ways to look at stuff. We did a very, very simple thing here. You can go much beyond what we did here, but I just wanted to kind of get this idea across. So what Luca did is simply look at IBD sharing in his data set. So the thing that he found was 2 observations. So first of all, he plotted this here in a way that looks at his data set. This is Southeastern Europe. If you don't recognize it, there's Greece down here. And of course, the landscape is cut. There's Croatia here and Romania and so on. And he plots now between these regions, he plots the average IBD sharing between individuals of, for example, this region with this region with this region. So he groups individuals into these regions and simply asks, how much IBD sharing do I have between these regions? You see this interesting web of connections And there's two observations here, and they are really simply summarized. That's why I like this kind of, why I wanted to get it across. There's nothing too complicated here. First observation, that I think you all see the first observation. What's the most obvious observation between Bronze Age and Iron Age that you see here? There's not less mobility, we'll come to that, but there's less connectedness. This is observation one, indeed. So there's a lot more connectedness in the Bronze Age than in the Iron Age, right? That's observation one. Observation two is far more subtle. And it only became apparent when Luca plotted the decay of this IBD sharing over space. So Luca now plots all of these pairs as a function of the distance of the pairs, right? So for example, between this and this region here, sorry, this and this region, you would get farther right on this, and for the close by regions, you would be here, and so on. And now you see something interesting. If we now model this decay with a simple squared exponential kernel, squared exponential decay curve, we see that in the Bronze Age, this decay may be much bigger. You see here on the y-axis, we're approaching 2 and 3 and so on, and this is 0.3 and 0.2, so about tenfold higher. This is observation one. But the second observation is that the decay is slower. You see a much larger radius, actually. So the way we interpret this is that in the Iron Age, people may have been less related, and I think the reason for that is much bigger population sizes. I will come to that in a second. But what we also see is in the Iron Age, people must have been related over larger distances if they were related. So what does that mean? I think that means that every person had a twice as big radius of interaction for some reason. So we'll come to that in a second, because there's another analysis that we did with this data set, and I'm kind of proud of, but it's sort of hidden in the supplement of this paper, but I should write it up properly. And that concerns another technology of IBD, also pushed by Harald, by the way, Harald Ringbauer, which concerns IBD within a person's genome. If you look at IBD, so common segments that are within a person, you measure relatedness of your parents. Okay, so it's called runs of homozygosity. If you have two same fragments from your father and your mother, that stretch of the genome becomes homozygous. It becomes exactly the same. There's no heterozygosity, no differences between these. So you can identify these, and it's the same concept. It's called IBD, sometimes called HBD, so homozygosity by descent. Sometimes it's called ROH, runs of homozygosity. Sorry, I'm putting out these terms here just for the experts that they know what I'm talking about. So and this expected runs of homozygosity is, of course, a signifier for recent inbreeding or recent consanguinity. For example, if you have parents that are siblings, you can simulate this now. If siblings have a kid, they would be largely homozygous here with this gigantic red bar. Sorry, there's no color scale here. These are the long fragments, and the blues are the very short fragments. But you can also show here. I can see that I need to round out this talk a little bit. This is the first time that I talk about this stuff, so apologies here. But the point is that there is also a signal of ancient population sizes in this data. So the smaller the effective population size, the more inbred everyone looks, but not in these kind of long fragments. So it's not recent inbreeding. It's not that everyone has sort of cousins as their parents, but you see in these short fragments that there is a higher rate of sharing in the past. So there's two observations. So there's an observation here that, there's a concept here that you can use this IBD within people to estimate populations sizes. And I built a probabilistic model, a Bayesian model that lets me estimate this as you go through time in the from here to here, this BC, or BP rather, so from 5,000 before present to 2,500 before present, Iron Age is about here. Sorry, I should use this pointer, otherwise people online cannot follow. So Iron Age is about here. And we can model this kind of rate of homozygosity with a simple methodology that was developed by Harald and Nick and others over the years that lets us draw a likelihood model of population sizes given this runs of homozygosity. And what comes out of this is this fascinating increase of effective population sizes through time, from going down from a few thousand individuals effectively, this is effective population sizes, going up by a factor 4 in the Bronze Age, then plateauing for a while, and then increasing again in the late Iron Age. So I find this fascinating, and there's a lot of stuff that will follow from these kind of analysis. I expect them to be more of those, and this is one of my major things that I'm currently working at. But Luca showing me this plot here came really as a shock, because I now realize something, and I have a kind of really bad slide for this, lots of text, but bear with me. So my idea is that there's a problem here. If people start traveling twice as far in the Iron Age than in the Bronze Age, they meet on average 4 times as many people. So in other words, the effective mating pool, the effective pool of people that you are interacting with increases by a factor of 4. And now, of course, it means that some of this effective population size increase that we've seen cannot be interpreted as an increase of population densities. It can be interpreted as effective mobility. And the shock here is that I was proud that we could kind of use this to make a story of, the naive interpretation of this kind of population size increase is, civilization thrived, people were productive, increase of population sizes. So clearly, no surprise, between the Iron Age and the Bronze Age in the last couple of thousand years, population sizes were increasing. Probably that is true. But it may also be the case that people just were traveling further. If everyone traveled further and further and further, we would get the same signal. So in other words, you can get the signal without a single change of effective population density. So this is where there are some links to theory. For example, putting forward by Nick Barton and others in 2013, but also Harald in 2017. So there's an interesting links here to spatial population genetic models. So in other words, the community has thought about this for a long time, but I am now reading up on this because I think we need to better understand the connection between mobility and effective population size. So that's why I wanted to talk about this briefly here, because it is really a conundrum that I'm thinking that we now have the data to look through time and space with ancient DNA, with these connections, but making sense of it is getting complicated because we have mobility and effective population size coming together and sort of amalgamating and producing the same signals. I hope that made sense. I wanted to get this across because it's an interesting link to population genetic theory, which I think has not been fully explored yet. There's a final chapter, and then I'm closing. Let me see. Oh yeah, I should end. The very last thing, and I wanted to put this in because it's another kind of super cool recent result that I'm proud of that we developed together with colleagues from the Natural History Museum who are here. I'm very glad. So this is a fantastic project that we put together from a micro region. So now we're doing the opposite as in the Balkan paper. So instead of looking at this European large region meta story, we're now looking at a super well-defined group here in Austria in the Traisen Valley. I actually traveled through St. Pölten, which I believe St. Pölten is somewhere. It's south. I see. Okay. I see. Okay. So anyway, this is the Traisen Valley, which goes into the Danube further north, and this was a very densely settled region in the Iron Age. So we're talking early Latène culture. So you see here a little bit of the dating information. So super confined data set, extremely well described archaeologically, extremely well defined anthropologically. We have evidence, we have data for many of these samples, like how old were they when they died. And there's just one last aspect that there's lots of that I could talk about this project. It's wonderful. So this is pushed by the primary analyst on the genetic side is Selina Karlhoff. And Selina has uncovered many interesting things about long range mobility, people who are outliers. How are these outliers integrated into families, like lots of stories there. But there was just one aspect that I want to get across today for this talk because it fits so well into mobility. One super interesting signal that we found is, again, a very, very simple thing to summarize. So Selina built these IBD networks and sort of computed, how is everyone related with everyone else? So we have lots of rich network data here that we can sort of play with. But one particular observable that I want to kind of draw your attention to is the overall rate of relatedness between pairs in the data set that are either adult-adult, adult-subadult, or subadult-subadult. So remember, this is something we can do with this project because we have anthropology partners who, for every single genome, gave us an idea like how old were they when they died. So this is for me something completely new. So Selina found an interesting signal here, which is that adults are related on average about 1%, and subadults are related on average about 7%. If you look inside of sites, if you say, we're not going to look at pairs between sites, we're just looking inside of sites, you see that adults are related 4%. So sure enough, if people are buried at the same site, they are more closely related, 4% versus 1%. But there's this massive increase of subadult relatedness, 80%. 80% of subadults on the same site are somewhat distantly related with each other. Now, if you look at this at first sight, you might say, well, that's not surprising. And why is it not surprising? Well, because adults are able to move more likely than subadults. So subadults are probably staying put. So if they die as subadults, they probably die where they were born. Adults are much more likely to travel. So what we could do with this is, with this difference, we could build an extremely simple model that told us what is the likelihood that people actually leave their place and are buried somewhere else. And this led us to two nice Bayesian estimates of lifetime adult mobility, which pointed us in the entire data set to something like 55, 60%. That is a large number if you think about it. It means that 60% of adults, 60% of adult-adult relationships that we should see in this entire region are missing. Why are they missing? Because people moved out of the region. It's a large number, right? 60% of adults are not buried where they should be buried, or if they didn't move. So they moved out. And the rate is even higher. It goes to almost 80% or 75% if you look at inside, so within sites. So 75% of adults move away and are buried at a different site in the same region, though. So that's the last story of mobility that I wanted to get across, because it's sort of a topological argument. It doesn't need spatial population genetic modeling like the previous one, or thinking. It's just a topological thing that we look at subadults versus adults. So I think that's a new concept that I'm quite excited about, because it's really something that we were only able to do because we drew together anthropology and genetics here. So just to remind you, these were the scales that we're talking about. Global human diversity very briefly, just to contextualize things, then these kind of continental and subcontinental stories about, for example, the Neolithic Revolution, but also then Iron Age versus Bronze Age, Southeastern Europe as an example case of how we can use population genetic modeling and mobility together to learn something about the human past. And finally, this kind of lowest scale, which doesn't really anymore rely on spatial concepts, but just topological concepts between sites and beyond. That's it. Thank you very much.